For those involved in the hair loss community, you've probably seen discussions about GT20029. Many describe it as being the most promising treatment for androgenetic alopecia that's ever existed, and I think I would agree with that statement, but a lot of people don't know a lot about this drug, and that's what I'm making this video for. The first thing I want to discuss is its mechanism of action and a general description about this compound. GT20029 is a PROTAC drug, which is a class of drug which allows for targeted protein degradation. What GT20029 allows for in a hair loss context is a completely unique mechanism of action for stopping androgenetic alopecia. GT20029 has been modified to target the androgen receptor, leading to its eventual degradation. In clinical trials, it's been administered topically in a gel form, which is important to note for later on in the video. The unique mechanism of action is why GT20029 is, in my opinion, the most promising upcoming hair loss treatment. Other drugs work by inhibiting DHT, such as finasteride and dutasteride, which alters systemic hormone concentrations and has led to side effects, however low they might be, in many users. DHT binds to the androgen receptor and causes miniaturization. And this process also led to another line of inquiry, topical anti-androgens, which bind to the androgen receptor and prevent DHT from binding to the androgen receptor. And these drugs are called topical anti-androgens. These include RU58841, fluridil, clascoterone or CB0301, and also pyrrolidamide. However, by degrading the androgen receptor, GT20029 stops the entire process involved in DHT-induced miniaturization from occurring. The cause of androgenetic alopecia, for those who don't know, is a process where a hormone called DHT binds to the androgen receptor in the scalp, and this DHT androgen receptor complex signals for the miniaturization of the hair follicle until it eventually stops growing. The way it causes androgen receptor degradation is as follows. The PROTAC molecule harnesses a system called the ubiquitin proteasome system to degrade a target protein, which is the androgen receptor in this case. This pathway is a natural process, and it's essential in your body, as the targeted degradation of proteins gets rid of proteins that are no longer needed in your body. Think of this as like a trash removal system. This PROTAC drugs treatment and line of investigation started because it seemed as a very promising treatment for cancer, as it targets cell cycle regulatory proteins whose degradation is vital for controlled cell division. These molecules consist of 2 ligands and 1 linker. One binds to the desired degradation protein, and one binds to the U3 ubiquitin ligase, causing the ubiquitinylation, or the addition of a ubiquitin to a substrate protein, which marks it for degradation via the proteasome. Importantly, after this process occurs, the PROTAC is not degraded itself and is recycled to continue this process for other desired proteins. In simpler terms, PROTAC drugs mark desired proteins for degradation and aren't destroyed in this process, allowing them to mark multiple desired proteins for degradation. This allows for infrequent administration. The ability for these PROTAC molecules to be recycled has incredible therapeutic promise, as classical inhibitors are limited by their one-to-one relationship with the desired protein. For instance, when IU588 is applied to the scalp, each molecule binds to one androgen receptor, thereby inhibiting DHT from binding to that androgen receptor. But in order to prevent inhibition of more androgen receptors, you have to introduce more molecules of IU58841 or any other topical anti-androgen, so the level of inhibition is dose dependent. This is important because by applying more anti-androgens to your scalp, you increase the chance of them going systemic and causing side effects. That isn't exactly the case with GT20029. Theoretically, you may only need to apply GT20029 a few times in order to get this inhibitory effect. This is seen in the phase 2 clinical trials. The unique qualities of GT20029 and its mechanism of action provides many advantages to previous hair loss prevention treatments in terms of efficacy, method of administration, and side effect profile. As a result, many in the hair loss community believe it's the most promising upcoming treatment, which again I definitely agree with. Due to its molecular size, it's believed to be very difficult to absorb into the skin when applied topically. This might sound like a bad thing, but we'll explore this later. It's actually very important when it comes to side effect profile, as its large molecular weight allows it to act entirely on the androgen receptor, or almost entirely, on the androgen receptor in the scalp. GT20029 is currently being studied and developed by Kintor Pharmaceuticals in China. Interestingly, the first PROTAC molecule was synthesized in 2001, but the first small molecule PROTAC was reported in 2008. So there was 7 years of development, and part of this PROTAC molecule includes a non-steroidal androgen receptor ligand, which for those who don't know is a selective androgen receptor modulator, or a SARM, which I just thought I would include because it's an interesting crossover between the hair loss and bodybuilding communities. Alright, let's look at the literature review, starting with preclinical studies and then going through phase 1 and 2 trials that Kintor Pharmaceutical has released on its website as like a press release. For the preclinical studies, a lot of the information was very difficult to find as it was published on Google Patents. According to one of Kintor's interim results announcements, GT20029 was used to treat acne in a testosterone propionate-induced skin hamster flank organ acne model. I know that sounds very confusing, but that was actually used to identify what the developmental name of GT20029 was in the Google patent. Looking at the filed Google patent by Kintor Pharmaceuticals, there was a patent titled Bifunctional Compound Preparation Method Therefore and Use Therefore, which mentioned a recently invented compound named A46. So from this we can know that this was the initial codename used for GT20029. Now, searching for A46 in the patent, we can find that the efficacy of GT20029 in promoting hair growth was tested in a mouse model of androgenetic alopecia using minoxidil as a positive control. The study found that both 1% and 3% concentrations of A46 were comparable in efficacy to 5% minoxidil, whilst a 0.5% concentration showed no statistical significant effect. As 1 and 3% concentrations were comparable in efficacy, you'll see that the evaluated dosage was 1%. These findings suggest that a sufficiently high concentration of A46 or GT20029 may be as effective as minoxidil in promoting hair growth. These efficacy results are needed for pharmaceutical companies to move into human trials. Now let's look at the clinical trials. In 2001, Kintel released a statement from the NMPA of China, which stands for National Medical Products Administration. Their statements showed that they had accepted its application for GT20029 to treat androgenetic alopecia and acne. Later that year, Kintel also released a statement that the FDA had given the IND, or Investigational New Drug Application clearance, allowing phase 1 human clinical trials to commence in both China and the United States. The phase 1 trials were successful in both the US and China. It's important to note, before we go further into this, that phase 1 human clinical trials are focused more on the drug safety profile and dosage ranges. The study in China was a randomized, double-blind, placebo-controlled study involving 92 healthy subjects. Both single and multiple ascending doses were tested. Using both methods help determine safety, tolerance, and potential effects before a full clinical trial. Phase 2 clinical trial. The results showed that GT20029 was safe and well tolerated, with limited systemic exposure. After a single dose, no drug was detected in the blood. Then, after 14 days of multiple doses, the maximum drug concentration was very low. It was less than 0.05 nanograms per milliliter. TRAE stands for Treatment-Related Adverse Events, and it's ranked in a 5-grade system. There were no treatment-related adverse events above a grade 1, meaning GT20029 was extremely well tolerated. The study in the United States was also a double-blind randomized, placebo-controlled study that had a parallel group design and included dose escalation. This study involved 123 healthy subjects, including healthy individuals and those with androgenetic alopecia or acne, as both conditions are androgen receptor related and are treated similarly in clinical evaluations. This trial found that GT20029 was safe and well tolerated with limited systemic exposure. In the single ascending dose stage, no drug was detected in the blood of healthy subjects. In the multiple ascending dose stage, the maximum drug concentration after 2 weeks was still very low, with the highest observed being less than 0.015 nanograms per milliliter. There were no treatment-related adverse events reported above a grade 2 in this study, showing that it was also again extremely well tolerated. It should be noted that the US had a larger sample size, so it's probably more accurate in elucidating the side effect profile of GT20029. Kintel progressed into phase 2 clinical trials with results coming out in April 2024. This multi-center randomized double-blind placebo-controlled trial enrolled 180 androgenetic alopecia patients across 12 clinical research centers in China. Participants were divided into a QD, or once daily, and BIW, or bi-weekly/twice-a-week dosing cohorts. Each group received a placebo, and experimental groups received either 0.5% or 1% GT20029 tincture, which basically means a gel form of GT20029. The length of the study was 12 weeks, and its primary goal was to assess non-vellus target area hair count, or TAHC. On a personal note, I believe this is the most accurate way of measuring hair count because vellus hairs are basically thin terminal hairs on your scalp, which are basically invisible unless you're measuring with a dermatological equipment, and they add almost no cosmetic effect. So this means that researchers were determined to not exaggerate their results, and shows that these studies were actually very high in quality. The 0.5% once daily GT20029 group experienced an increase of 16.8 hairs per centimeter squared from baseline, which was 7 hairs per centimeter squared more than the placebo group, which was statistically significant. Additionally, the 1% biweekly GT20029 group showed an increase of 12 hairs per centimeter from baseline, exceeding the placebo group by 7.36 hairs per centimeter squared, which again was statistically significant. This ultimately indicates to me GT20029 is extremely efficacious. The article notes no adverse sexual events were reported, indicating if GT20029 makes it to the market, it will probably be advertised to be a more tolerable compound in terms of sexual side effects compared to finasteride, in order to create some product differentiation. For those who experience sexual side effects from finasteride or dutasteride, this is a very promising treatment for you. Based on these findings for phase 2, it's likely that the 1% bi-weekly dosage is optimal and will be used in subsequent phase 3 clinical trials. Now I want to have a discussion on its safety profile, add my own insights from everything I know about this drug, and what could be expected when it hits the market. As elucidated earlier, GT20029 works by degrading the androgen receptor, eliminating DHT-induced androgen receptor signaling that leads to the miniaturization and eventual death of the hair follicle. However, this begs the question, does GT20029 go systemic when applied topically? And what kind of side effects could be expected if this was to take place? As discussed in my article about RU58841, this discussion of topical drugs going systemic is a bit more complex than how it's described typically in hair loss forums and communities. It's extremely hard for a compound applied topically to have any systemic absorption, because at the end of the day, the way it's able to work and introduce an effect is by absorbing through the skin. It's applied topically to get the most effect at a localized area, which in this case is the scalp, but it's basically inevitable for some amount, however small, to be absorbed systemically. The reason for a compound causing systemic effects is predicated on things such as molecular size, binding affinity, half-life, and IC50, which stands for half maximal inhibitory concentration. Also, these factors are for its resultant metabolites, because once whatever the compound is broken down by your body, the metabolites might exert a similar effect. So it's also important to consider the effect of its metabolites. The first factor to discuss when questioning GT20029's ability to go systemic is its molecular size. As discussed before, there is very limited information about GT20029 available. However, looking through the patent, the liquid chromatography mass spectrometry result is listed for A46, which, by the way, is its initial research name for GT20029. The molecular size of GT20029 is notably high at 1043.2 daltons or grams per mole. For those familiar with the 500 dalton theory of skin absorption, this may seem to mean that it's almost impossible to be absorbed when applied topically. However, there are a few things to consider. Firstly, the 500 dalton theory isn't widely approved, as there isn't a huge body of evidence to support its validity. Instead, it's more of just a guideline used by pharmaceutical and cosmetic companies when testing their products. Additionally, there are now enhanced delivery techniques such as liquid and gel carriers that allow topicals to bypass the skin barrier, and also even things like humidity can affect skin absorption. This is all very individual, and your skin chemistry can affect absorption too. With all that being said, it's clear GT20029 is absorbed through the skin because, like we've seen in the phase 1 and 2 clinical trials, it exerted a statistically significant effect on the hair follicles and was also tested to be found in the bloodstream. So, I mean, it's clear that it was actually well absorbed through the skin. Its high molecular weight is actually advantageous in this regard, as it acts as a barrier for excessive absorption, so the remaining molecules which actually make it into the system will act almost solely on the androgen receptor. This is similar to how dutasteride mesotherapy is described to work without going systemic, and this is shown in a lot of blood work results. Keep in mind, this is far from an exact science. The second factor to consider is GT20029's half-life, IC50, and its persistence systemically. As mentioned, phase 1 clinical trials focus more on safety data, and looking at the US group, it was said that the maximum drug concentration after 14 days was still very low. Considering the total amount of androgen receptors, this is unlikely to affect the totality of the system and androgen receptor mediated functions such as muscle mass, strength, bone density, and sexual function. However, this is just speculation by me. It's not clear what the half-life of GT20029 is, but due to its unique mechanism of action, it doesn't need to be administered daily, which was also confirmed from the findings in phase 2. And it's more optimal in a bi-weekly protocol. This gives time for the drug to be metabolized or broken down by the body, so by the time of the next administration, there are no levels persistent in the bloodstream, which allows for the drug to not accumulate to any significant levels in the serum, thus not affecting the totality of androgen receptor mediated functions in the body, such as muscle mass, strength, sexual function, and bone density. This is the reason why topical finasteride treatments don't negate side effects, because since finasteride has such a long half-life, when inevitably that small amount of finasteride goes systemic when applied topically, by the time you apply it the next day, it's still present in your blood. So when you take it the next day, the amount increases, and eventually it will accumulate to a systemic. It will accumulate systemically enough to affect DHT levels in other parts of your body, such as the prostate, and that's why topical finasteride doesn't negate side effects compared to the oral form. Alright, ultimately, I think GT20029 is the most promising step towards curing androgenetic alopecia, and it takes a unique step forward, which I haven't seen in, a very long time since topical anti-androgens were evaluated in the late 90s. Clinical trial data indicates that even when minimal amounts go Systemically, their levels are extremely low and are unlikely to disrupt systemic androgen functions. The biweekly application protocol further mitigates risks by preventing accumulation in the bloodstream. Thank you for watching, and I'll see you in the next video.